Hi guys, today I'll be going over the basics of the genetic duplication process known as PCR. So what exactly is PCR? PCR is a fairly recent procedure that allows for breakthroughs of scientific advancement. It provides easier access to DNA sequences with the ability to copy one specific segment to make millions of copies in only a few hours. As opposed to previous methods used before PCR was invented, such as DNA cloning, PCR is much more efficient because of its reduced time requirements. And plus, it's a lot cheaper to use as well. The PCR process is critical in genetic identification when only small amounts of DNA are provided to analyze and compare to other samples. This copying of DNA is found to be useful with not only identification, but also genetic modification and research in terms of pathogenic identification as well. It's a simple process that can be performed in a high school laboratory setting without costing too much money or time. PCR itself occurs in 3 phases, which repeats over and over again for as much as 30 cycles. The first phase, or denaturation, is the process in which the double helix structure of the DNA is separated into 2 single strands. So if we go back to this diagram that we had before, you can see that right here, before the process begins, we have the original DNA strand, and right here it is being denatured in step 1, or split into half. The reason why this can occur is because PCR is a heat-sensitive process, so when the DNA is brought to higher temperatures, say between 94 to 96 degrees Celsius, it splits apart into 2 strands because of the nature of DNA. And here in this previous slide, you can see right here, this is known as a thermocycler, and in the process of PCR, it can be used to moderate the temperatures that the DNA will have to go through, the changes that it goes through. The next step in the process is known as annealing. The temperature of the DNA is brought down to 50 to 65 degrees Celsius. This is optimal for primers, or short nucleic acid-based sequences, to bind to the complementary denatured strand of DNA. So this would be step 2, right here. So in other words, primers combine the nucleic bases, or adenine, with thymine, guanine with cytosine, and together they form base pairs on the denatured strand. The primer is only around 10 base pairs long, but it will be extended in the next step. The final step in PCR is, you guessed it, extension. The primed complementary strand with only 10 base pairs is extended to the end of the sequence with the help of an enzyme known as a DNA polymerase. One of the most commonly used enzymes is known as Taq polymerase, spelled T-A-Q. Taq polymerase, which is helpful since it's a heat-resistant enzyme that won't change with altered temperatures. This enzyme in particular works at an optimal 72 degrees, where it attaches to a complementary strand and extends outwards. So this would be step 3 right here. So here we have the denatured strand with the primer, and the Taq polymerase attaches to the primed strand and it extends it outwards. So all the way outwards until there's no more DNA left to replicate. And this would occur over the process of like 30 cycles to create millions and millions of copies of DNA. Here's an animation that I found online that might help with your understanding of the PCR process. This is how it would look in a laboratory setting. So we have a tube filled with the DNA polymerase, the sample itself, the nucleotides, adenine, thymine, guanine, cytosine, and then you have your primers. And so we add this tube to the thermocycler and begin the process. This is how PCR looks like up close. So first, here we have the target sequence on the DNA sample, and as the temperature is raised, it unwinds and denatures. And then the temperature is lowered again, and the primer binds on to complementary sequences adenine with thymine, guanine, cytosine. Then, as the temperature is raised to 72 degrees Celsius, the DNA polymerase binds on, and with the nucleotides in the solution already, it extends it. So let's watch that. And so, as you can see, by the end of the first cycle, you have 2 separate strands, which are going to denature once more. The primer is going to bind on, polymerase will extend it on, and see, by the third cycle, These are two of the desired fragments that you originally wanted to replicate. And by cycle 4, it's become 8 fragments that you wanted to replicate. Now let's see what happens over the course of, let's say, 30 cycles. This process takes only a few hours, and as you can see, billions of fragments have been made from the original sequence that you wanted to make. This is useful in the replication of DNA that's used every day. I hope this video was informative, and thank you for watching.